This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 76, recorded on April 10th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How have you been?
Not too bad. I'm teaching medical students right now, so I'm refreshing my memory on a lot of interesting biology.
Do you do a lot of lectures for them?
It depends on how you define a lot. I'm doing 7.
That's not bad.
But 4 in 4 days at 8 a.m.
Yeah, that's God. A problem. Ooh. It's early.
It's you have to get used to it.
Yeah, well, it's the way. I know. It's the same here, and I have to get up really early because I have to drive a long way. And if I get stuck in traffic and turn up late, that's no good, so...
Yeah, it's just a short bicycle ride for me.
Nice.
Because the snow is gone.
That's right, it is. It's spring.
Yeah.
Also joining us today from Small Things Considered, Elio Schaechter.
Hi there. How are you?
On the West Coast where it's always spring, right?
Absolutely.
No snow. Have you had rain?
No, we had a little bit of rain, and it's a disaster because we're really in a drought condition, and that's serious.
Yeah. It's a big problem. Yeah, a lot of our food comes
mushrooms.
from California.
Yeah.
And that's not good. Also joining us today from, I should say, Charleston. Is it Charleston?
It's Charleston.
Charleston, South Carolina. Michael Schmidt.
Hello, everyone.
I usually say Medical University of South Carolina, but I want to say Charleston now because that's how everyone else gets. Yeah. I should write it down. Otherwise, I'm going to forget. How are you, Michael?
I'm doing better. I'm almost through the bronchitis. It's been an ordeal. It's been going around the city. And if some of you saw the Today Show on April 10th, they were talking about this new study out of MIT talking about how far a sneeze goes. Yeah. 200 times further than we ever thought. So Matt Lauer brought that to everyone's attention, and as many of you know, Matt Lauer is a germaphobe, so he was quite concerned, but he passed on the great public health message of covering your cough and sneeze so that you can limit the spread.
In the crook of your elbow, right? You're not supposed to use your hands.
Crook of your elbow.
You're spreading the germs through your hands.
That's right.
Oh, so I'm sure some of the sneeze still gets out when you put it in your elbow.
Of course.
Oh, yeah.
So what, do they have some new tech that can see the droplets going farther?
Yes. MIT used the term, they had a fancy term for it. It was turbulent disruptive something or other. As I was drinking my coffee this morning, half listening to it, but my ears perked up, and it's... turbulent, where did I write it? Oh, multi-phase turbulent buoyant cloud. A multi-phase turbulent buoyant cloud. And they can understand why I needed more coffee to remember that.
Well, even when you talk, you make aerosols, so I wonder how far they go.
Yeah, as before we came on, I was at seminar, and we were talking about that Today Show piece before the speaker started. And one of the interesting things was I said, well, it's sort of like an R naught. How many people are infected by your particular sneeze? And, you know, the R naught concept is how many infected people infect downstream. And for things like mumps, which is an airborne disease, it has an R naught where one person infected with mumps will infect between 12 and 18 others. And while influenza from 1918 that we all remember only had an R naught of 2 to 3. So it's another public service message to get your kids vaccinated. And one of the things that we're having a big problem with is measles.
Yeah,
and
We have an outbreak here in New York City.
And it's got an R naught of between 12 and 18 as well.
It's one of the high ones, yeah.
Yeah.
We've had 20 cases here in New York, mostly in people who have not immunized their kids. Talked to my class yesterday. I said, you can get around certain vaccinations for philosophical, medical, or religious reasons. And I said, I don't understand the philosophy of wanting to get sick.
That's true.
Doesn't make any sense. All right, we have two very interesting papers today. And the first one is published in Nature. And it has the title, which you may find unusual for TWiM, Sensing Array of Radically Coupled Genetic Biopixels by Prindle, Samoya, Raznikov, Danino, Simmering, and Hasty. Oh, these are all in La Jolla. And
That's right. They're all at the University of California, San Diego, all associated one way or another with bioengineering, bioinformatics, and the biology department.
How did you stumble on this, Elio?
Well, I have a story about this. Jeff Hasty, the PI, lectured in a graduate course that I helped organize, and I'd like to tell you a little bit about the graduate course because A, it gets me in touch with fabulous stuff like this, and B, since the paper is at the very edge of what I can handle, I had, luckily, two of the graduate students in this course, well, actually, one in this course and one a friend of his, wrote a piece for our blog, Small Things Considered. And, of course, being young and bright and all that, they understood it. And I am just going to be parasitic about this, and I'm going to use material from their write-up, which helped me understand the paper. So Iman and Alex, I apologize to you if I'm being a little bit, what's the word? I mean, parasitic is the right word, I guess. But you did such a good job that I will be... I'm in the clear by using your material. And for anybody who wants to read it, it'll come out on April the 28th in Small Things Considered. But let me say, if I may, I'd like to really digress. This is a big-time digression, and tell you something about this graduate course, which we have been teaching at University of California, San Diego, in cooperation with San Diego State University, for about 12 years. It's a small course. It's a graduate course designed for beginning graduate students who don't know much microbiology. And the characteristic of the course is that instead of regular lectures, we bring in, for every lecture, a guest. And the guest is supposed to sell to the students why hers or his are the queen of the sciences in 40 minutes. And then we discuss a paper. And the result of this is that we do not worry about coverage particularly, although we kind of do. We sort of try to go into the far reaches of microbiology. It's a 2-quarter course, by the way, so it goes from January till the beginning of June, and we meet twice a week for an hour and a half each time. Anyhow, what is so unusual about the course is that there is no professor. There are scientists talking to fellow scientists. And the result has been a degree of participation and excitement that you would dream of. Okay? The students feel they're being treated like grown-ups. We feel we're not there to enforce anything. We are there to share in the excitement. And let me give an example of what happened. Last year, for a final, and the exams are, of course, take-home exams, write a short grant application about some such and such a subject. But last year, we decided, instead of that was kind of Mickey Mouse. It was still too didactic for our blood. So we said, the exam consists of the following question: write an essay on a subject of your choice. Okay? An invitation to goof off if you want it. And out of 16 students, not one did. In fact, 4 of the papers, I remember, were so in-depth that I had to enlist the help of colleagues to evaluate them. And another 4 made it into our blog, small things considered. This year, in the after the winter quarter, we decided that instead of a final exam, we're going to ask them to write for the blog, and to do it in pairs or groups of 3 or something like that, so that in addition to everything else, they learn how to cooperate, how to work together. The result of this has been stunning. I mean, they can't wait to do it. And the participation in the class, as I say, is a level of excitement that I cannot describe to you. It is truly amazing. And so what my role there is simply to organize the course together with one of 2 professors at UCSD. In the winter, it's Joe Poliano. In the spring, it's Doug Bartlett. It's Scripps Institute of Oceanography. Anyhow, it's a great experience. So that's why I'm leaning on this paper by Eamon Riley, who's a student at UCSD, and his friend Alec Miske. Alex is a graduate student at Harvard. So this is an intercontinental cooperation. Enough of that?
Sounds good.
Can I start on the paper?
Yeah.
Okay. So what's the paper about? It's about making biosensors. What are biosensors? Biosensors are using living things to amplify a signal. Okay. So if you use biosensors, especially bacteria, you can detect very small amounts of chemicals and potential toxins. You can rig this to detect pollutants in water, contaminants in food. You can use this to diagnose medical conditions, and I'm sure that the right people are involved in using just this kind of technology to detect agents of biological warfare, for instance. So it's a big subject, biosensors. So a biosensor is a way in which a microbe senses something, responds to the chemical information, and is engineered to transmit a signal. Biosensors tend to be low cost, they tend to be very portable, not a big gadget involved, and they are highly sensitive, and they can register what's going on in real time. So they obviate the need for expensive equipment and laboratory work on a large scale. So now the way they work usually is by looking at an oscillating feedback in response to a signal. Now, oscillations are something that excites engineers very much, and bioengineers are no different. They love oscillating systems, and one can build a whole lot of them, and a bunch of labs have done it. There are people at Caltech, at Rockefeller, at the Weizmann Institute in Israel, and of course here at University of California in San Diego. Many other places work with these things. It's a very popular subject among bioengineers. And to understand it, you have to really be familiar with the way circuitry works. And I already said that. That's not me. Okay. So anyhow, what happens is that one of the troubles with using biological systems is they're noisy. There is a variability between one cell and another, and this can muck up the signal. So one way to do this is to use large-scale synchronization of individual cells. If you take a culture of bacteria, or if you took a paste of bacteria or anything like that, a biofilm, you find that it's very heterogeneous. But you can line them up in a way by using the right genetic ways. And the way that is favored is to use frequency-based circuits, which are said to be much more robust than amplitude-based. And I'll remind you that the abbreviations FM and AM refer to exactly that. That's what we're talking about. So frequency modulation has it all over. Amplitude modulation in an FM radio sounds a heck of a lot better than an AM radio. Isn't that right?
Absolutely.
That's absolutely correct. Fidelity.
Okay. Fidelity. And that kind of fidelity is the basis for, of all things, AC power, alternating current power, GPS, lasers, and who knows what all. And it's attractive to build that in to a biological system, namely bacteria. By the way, I'll anticipate where the paper is going. They succeeded in doing exactly that, and they put it to use to detect contamination by arsenic in water. So they already have a proof of principle experiment in this paper. The paper, by the way, came out a couple of years ago. We usually try to discuss newer papers, but this one is such a trend-setting one that I think it doesn't matter if it's been out for a while. Anyhow, bacteria communicate very well on a local scale. One bacterium talks to another bacterium, and that's it. But they have a hard time talking to each other at a distance. And part of the problem is, and of course, the granddaddy of all communication systems is quorum sensing, which depends on the density of bacteria in a given volume. So, briefly, what this means is that bacteria secrete certain compounds which are inducers, or they're signals, the signaling molecules. They can induce a genetic system, and this happens in a variety of systems having to do with virulence, having to do with bioluminescence, and so forth. So, this is well known, but this depends on the diffusion of chemicals from one bacterium to another. And I remind you that diffusion is relatively slow on a macro scale. The cup of coffee that Michael just drank, if you put sugar in it, he stirred it, didn't you?
Absolutely.
Okay, and if he hadn't stirred it, what would have happened? It would have taken a couple of hours, if not a day, for the sugar at the bottom to equilibrate with the rest of the liquid. So diffusion is, from this point of view, is slow. So the lab by Jeff Hasty came up with a way of rewiring this and adding to it not just quorum sensing as a signaling mechanism, but also a gas phase. Now, diffusion through gas is much faster, and that becomes a much better way of communicating. And they did it by making it possible for the bacteria, in response to a signal, to produce, of all things, hydrogen peroxide. Now, hydrogen peroxide is water, essentially, and it vaporizes. So it comes out because they will have a system which is not closed. It doesn't have a lid on it. So hydrogen peroxide will come out and it will diffuse throughout whatever you want to diffuse. Okay, so that's the basic idea.
Let me ask you one question, because that really intrigued me, is the whole gas business. What was their thinking behind making a gas? Because wouldn't you have to make more to account for it moving away in order to get their FM effect? Wouldn't it,
sure,
like, you'd need more? And so did they share any of that wonderful insight as to how they came up with the concept of gas?
I think it was because of the speed with which gas diffuses.
Okay,
they knew
so
that
they did a trade-off.
They did the trade-off, but also they enclosed. We're talking here about their test tube is not a regular test tube. It's not an Erlenmeyer flask. It's a microfluidic device. So things are done on a small scale. And although there is a gas phase to the gadget, it's covered. So it doesn't just the gas will not diffuse into the room. It will stay within a certain volume. That helps.
Crystal clear.
Okay. All right. So we're talking here about two systems. One, quorum sensing, which works very effectively, but over a short distance, and another one which may work, as you point out, a little bit less effectively, but over large distances, namely over a gas phase. By the way, how they came up with hydrogen peroxide really blows my mind. I mean, I would not have thought of that. I would have thought of some gas that is typical of bacteria, that H2S or something like that. But they used plain, ordinary hydrogen peroxide as the sensing. So this gets a little bit complicated when you think about how they did it. But before I tell you how they did it, they arrayed this into a bunch of colonies over little tiny squares in their microfluidic device. And they call these biopixels. So they're essentially bacterial colonies, okay? And they all carry the same genes, the same constructs, and they all, because they are in communication through the gas phase, but not through the liquid phase, they will eventually become synchronous. And this phenomenon is called synergistic synchronization. It requires the synchronization via quorum sensing and via the gas phase. And before I tell you about it, Jeff Hasty came to our class and he showed a video. And this is a video clip from somebody in Japan, laboratory of Professor Ikeguchi, and he showed the following. Picture 32 metronomes, tick-tock, 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 right? And he puts them on a table. The table is magic. It's sort of a table that sits on a couple of rollers or surface. Anyhow, there are 32 metronomes, and he gets them started, and they're all asynchronous. Cluck, 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 cluck. Okay? And then after a while, you notice something is happening. Within minutes, they start to move synchronously. So pretty soon, instead of clack, 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 you have clack, 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 clack. I'm not very good at that, but you can picture that. At the end of 2 minutes, they're all synchronous. Okay? It's magic.
And if you put it on a fixed platform, this doesn't happen, right?
That's right. If you put them on a very heavy platform, it has to do with the fact that the platform induces some signal that is picked up by the metronomes. So that is magic, okay? And actually, anybody can look at this. If you go to YouTube, just enter synchronous metronomes, and it pops up right away, okay? So you don't even have to give people a guide as to where to find it. Anyhow, the same thing happens with this bacteria. And the signal here is not the movement of a metronome, but light, because the signal that is eventually emitted is in the form of bioluminescence. So it's simple to pick that up, and pick up they do. So how does this circuit work? And by the way, the number of biopixels is high, so 12,000 biopixels. So you're talking about 50 million cells. Explaining the genetic circuit is a little bit complicated. It involves several modules. There are 3 interconnecting modules. One is a quorum sensing module, which encodes for the enzyme that makes the quorum sensing molecule, which here is acyl homoserine lactone, AHL. So this one module has, first of all, it has a promoter that controls the synthesis of AHL via its synthetase, but it also has, under the same control, an AHL degrading lactanase. So this becomes an oscillating system. Under the control both of hydrogen peroxide and LOX-R. LOX-R being, it's well known among microbiologists. It's a transcriptional activator that drives the expression of the genes that are here. One makes the lactone, the other one degrades it. Now, this becomes a self-reinforcing oscillating system because the inducer induces the formation both of its own self and its own degradation. This is restricted by diffusion. Okay, this depends on AHL, acyl homoserine lactose. So it's restricted only to what happens within a pixel. To make the pixels communicate with each other, these colonies communicate with each other, they use this H2S business, and they do this by encoding NADH, the hydrogenase, which, for those who remember the biochemistry, generates hydrogen peroxide, which becomes a vapor. And eventually, and this is a bit more complicated than that, eventually this leads in synchronized oscillations in hydrogen peroxide levels, which allow rapid transmission of signals over large distances. And so the hydrogen peroxide diffuses over the array, and everything eventually becomes synchronized and can be read out with a photometric readout for light. Okay. I don't pretend that this is obvious from listening. So for the readers who are not engineers, it may pay to go to the paper. So now, what good is this? So they put it to work to detect a common water pollutant, arsenite. Arsenite is a problem in many parts of the world. Michael, you probably know about this. It's in India, isn't it, that this is a big problem, in Bangladesh also?
Bangladesh as well.
Yeah.
This is why I think it's such a phenomenal thing that they picked arsenite as their indicator, because we need a biosensor that's really cheap, because you can't put a mass spec or one of these other very sophisticated instruments associated with these wells. So I think they've done a tremendous service for the world.
Right. I think that's correct. And I think they have data that shows that the frequency oscillation is altered. At this point, just let's take it on faith that they have a construct that is arsenite responsive. It deals with a promoter that can detect arsenite as well, and is coupled to the quorum sensing response. Anyhow, let's just pretend that we understand that and go on. And this then allows to detect very small amounts of arsenite very fast. And you can play around with this. So they play around with this by using different distances between the arrays, and this gives them some information about, it can be used to maximize the condition. So these are inexpensive and responsive biosensors that could be used in developing countries. They could be used by the military. They have advantages in testing. And like you say, they don't require big machines. And so they have come up with this really rather remarkable paper.
What is the readout in the end that you're...
Light.
It's light, so you're measuring light as a...
Right, and that's cheap.
For the presence of arsenite.
Right. That's right. In the presence of arsenite, you get this bursts of light, and you can record them. That's not expensive to do. And by looking at the pattern, you can determine the amount of arsenite.
And practically speaking, if this is cheap, you would assemble it into some kind of container, I presume?
A portable container, and they show you a diagram of a portable container in the paper, if you want to.
What's the shelf life? Once you've loaded the microfluidic device, how long are those bacteria stable?
Wow, that's a good question. I'm sorry, I don't know. But there's not a lot of information about it. They talk about portability, and maybe in the paper someplace it says, but I didn't see it.
Well, if they're generic bacteria, you can probably load them up on the device, and if they synchronize as quickly as the metronomes, and they probably do because of the genes making the gas, you could play the game. And I was so intrigued with this paper, I went out hunting to see if Dr. Hastings' group made a YouTube video like all good labs do these days. And sure enough, there's a YouTube video out there, and I posted it for our good friend Ray to put in the show notes. And you can actually see the video of the synchronized bacteria that I'm sure the graduate students showed you in your exciting class.
Professor Hasty did. He came to the class. He lectured to us. Yes. Also, the paper has in the supplemental material, there's a bunch of movies. So it's there as well.
So as long as these promoters are active in stationary phase cells, that would increase the shelf life, I would guess, of these devices.
I would think that you can do this over a period of maybe a week, something like that. I'm guessing.
Then you have to,
I would think so.
Then you have to replace the bacteria, which is not a big deal.
Probably it's not a big deal. No, it's modular, so you just insert another cassette. I mean, you don't mess around, you know.
And then you could design this to sense any number of things that could be sensed by the bacteria, basically, right?
That's right.
And anybody who's really fascinated by this, one of the students in my group pointed out that the Hasty group just had a new paper in Nature where instead of using these molecules, they're actually using the clip protease, which is highly regulated.
Much faster.
Yeah, it's much faster. And then the idea is when the protease clips a protein, you release, you get light.
That's
you
right.
activate light.
Yeah.
So
I was going to bring that up, but folks, this just came out yesterday.
right.
It takes me a little while to digest papers like this, so
Right.
I didn't do it. But yeah, thank you for mentioning.
The creativity
Michele.
is really impressive.
It's very impressive, and the figures are just remarkable. I mean, It looks like it's done by really high-class engineers who know exactly how to produce exact measurements.
And I think we should emphasize that they're taking advantage of decades of really mechanistic research that went into identifying these circuits, these signaling circuits, and the mutants that they use to prove their concept. So it's a nice application of a lot of beautiful basic science.
So Hasty is a microbiologist, correct?
Hasty is a bioengineer.
A bioengineer.
I don't know his background, but I don't think he's trained as a microbiologist. But the beauty of coming in from another field is that you don't have any restrictions about your habits. You know, we are all in the habit of using systems we're familiar with and so forth. If you come into a new field, if you became an astronomer, Vincent, you wouldn't have any hang-ups about what system to use, right?
Yeah.
You go for the jugular. And this is what they do. They go for the jugular.
But they created a new department. This is the first time I've ever read a paper from a Biocircuits Institute.
Sounds cool. I like it.
They have a whole collection of people. First of all, let me say that major research universities are going very deep into bioengineering. This is a habit that they have. It's certainly the answer to, is your university doing something useful? You know, and everybody's trying. And so bioengineering is really in the forefront of this. And what it is, it's a long way from just people who make, little gadgets. These are very, very, very sophisticated people who work together with biologists. So,
as Michele said, though, there's a lot of basic research here that makes it work, and we can't stop doing that. Otherwise, the future biocircuits are not going to work.
That's true.
We still
let me ask the group this question: do you think that this sort of technology will become as ubiquitous as GFP has become in biology today, which is the green fluorescent protein to do readouts of everything from neurobiology to, every paper you read has a variation of green fluorescent protein cassettes in it to detect things. And I think the GFP system was the first real biocircuit that saw widespread use.
I'm not so sure. I think that the bioluminescence, the LuxR system, has been around for quite a while, and it's been used quite a bit. So there are at least two systems which are widely used, and which one is more convenient depends on your gadgetry. But there's a lot of bioluminescent stuff, and it has been used just as a reporter system since the early days.
I think GFP worked, Michael, because it's pretty straightforward for most molecular biologists to use, right?
Yeah.
It's a matter of putting a gene in an organism and having a UV microscope. So that's a requirement that a system has to be simple enough to get widespread use among people who aren't experts, right?
That's it.
So that's amazing. This is really a first for TWiM. We haven't done anything like this before. Well, thank you, Elio. And thank you, students in the course.
Oh, especially thank them. I'll give you the name once more.
Yeah.
Eamon Riley and Alex Miske.
Nice. Now, the one is from Harvard, right?
Alex is from Harvard, and Eamon is from University of California, San Diego.
So how did he take the course at Harvard?
No. The Harvard guy is not taking the course.
He just helped.
No, he's just a friend. And this is a nice thing. I like that. I mean, Eamon came to me and says, "Listen, can I work with a friend at the distance?" And I said, well, communication at the distance is what this paper is all about.
Yeah.
So you might as well.
Good answer, Elio. I like that. All right. Let's move on to our second paper today, which is a letter published in Nature. It is called Microbiota-Liberated Host Sugars Facilitate Post-Antibiotic Expansion of Enteric Pathogens. And I have to say, personally, I could understand this title a lot better than the title of the first paper. But I do like the name Biopixels. That's pretty neat.
Yeah.
The authors here are Ferreira, Higginbottom, Lynch, Kashyap, Gopinath, Naidu, Choudhury, Choudhury, Weimer. I'm probably pronouncing them all wrong. Monak and Sonnenberg. And they're from Sonnenberg. I know enough Sonnenbergs to get that right. Here it's from Stanford, also University of California, San Diego, also University of California, Davis, and the Mayo Clinic in Rochester, Minnesota. So this is all about understanding when you take antibiotics and you disturb your gut microflora, why pathogens can move in. So we know that antibiotics disturb the communities that are the bacterial communities that are in us, and this leads us to being infected with pathogens that normally are not there. And there's a word for this. Protection by the microbiota is called colonization resistance. And people have been trying to understand how this works because there are specific mechanisms involved. It's not just a matter of having a niche occupied, but there are functional mechanisms behind that. And one of the ideas is that you have all these bacteria in your gut. There's some that degrade carbohydrates from a variety of sources like fibers and starches, and then those carbohydrates are made available to other bacteria, and they make short-chain fatty acids, which we've talked about before in terms of their effects on colon cancer and inflammation. And basically there's a food web in the gut that depletes nutrients, and therefore invaders that require the same nutrients can't compete. You can't just step in and say, give me your food, because they're not going to give it to you. They're going to keep it for themselves.
It's a little bit surprising. Although I'm sure the statement is correct that there is depletion of nutrients, what comes out in the form of feces is actually fairly rich in food still. If you were to make a suspension of feces and filter out the solid material and the bacteria, you could grow bacteria in it.
Well, maybe it's not the right food for the invaders.
It's not. It's right. It's probably right. Yeah.
Yeah.
Well, which you're going to probably get into, Vincent. You're going to tell us of how they made mutants and all those other wonderful things.
I absolutely will. This is a wonderful combination of systems biology, where you look at big data sets of genetics, fundamental bacterial genetics, a little metabolomics, carbohydrate chemistry. And that's why we have all these people on the paper with different expertises, I'm sure. So in this idea that there are nutrient niches in the gut, and these prevent infection, this has been shown in mice before. So, for example, if you take mice, you treat them with streptomycin to deplete or change the gut bacteria, you can then colonize these animals with enterohemorrhagic strains of E. coli. All right. And then and they will grow and take over and cause disease in the antibiotic-treated mice. But if you instead put commensal E. coli back in the mice, and these E. coli are now using the sugars that the enterohemorrhagic strain requires, you will establish colonization resistance. In other words, the commensals will prevent the colonization by the hemorrhagic strains. And similar experiments have been done with Clostridium difficile, which again is one of the bacteria that can come in, one of the pathogens that can come in or can take over when antibiotics are treated. So essentially, diversity of the microbiome is really important for this kind of resistance. So that's the background as I understand it. And what they do in this paper is to try and understand the mechanism. Why is there colonization resistance? They use Salmonella enterica serovar typhimurium, or Salmonella typhimurium, and Clostridium difficile to bacteria that are often involved in antibiotic-associated disease. They also use mice, gnotobiotic mice, that is without bacteria in the gut, and then they colonize them with Bacteroides theta iota micron, which is a gut microbe. And now, so they make basically what they call a reductionist system. You have a very simple microbiota in these mice, and then you ask what's important for C. diff or S. typhimurium to come in. So I'm gonna abbreviate Bacteroides theta iota micron as BT, even though it's going to confuse. You could confuse it with BT, which you throw on your lawn to kill bugs, but this is different. It's a common colonizer of the human gut, and this bacteria is able to degrade polysaccharides that we either eat or which are present in the mucin layers that are coating our intestinal tract. So in one experiment, they have mice that are either gnotobiotic or colonized with BT, and then they put in S. typhimurium, and then they take it out after a certain amount of time, and they look at the transcriptome. They want to know what genes are turned on in Salmonella. And basically what they find is that Salmonella genes that are involved in catabolizing sialic acid and fucose are upregulated when BT is present. And these are sugars that are typically found on carbohydrates. So they're usually terminal sugars. Now, BT is able to cleave these sugars, sialic acid and fucose, from the ends of sugar chains. They have a sialidase, and that, for example, that takes off the sialic acid, but BT doesn't degrade the sugars.
And these are sugars that are on the lumen of the gut.
Right.
So these are human, or in this case, mouse sugars.
Although we have them too.
Yep.
Yeah. In fact, sialic acid is the receptor for influenza virus when it's present in the respiratory tract.
So they're scavenging these from the sugar coatings of our intestinal tract.
They are. It's a lovely thought.
That they're scavenging them, or I almost...
I leave it to you to figure it out.
I actually think it's a commensal relationship, that they're actually serving as the microbes are serving as our first layer of defense from the evil ones that are trying to get into the sterile body tissue.
Yeah, possibly, yeah. That makes a lot of sense, right?
And in return, they eat our sugars.
Yeah.
Well, they keep our weight under control. That's the way I look at it.
So one thing I found interesting, this B theta, which is a bacteria that lives in our gut, they make a sialidase that cleaves sialic acids off, but they don't degrade it. And they noted, they make a comment about this in the paper. They say, well, it's not clear why they would do it. Maybe it's so that they can access the next sugars in the chain of sugars. Sialic acid is the terminal one. Maybe they're taking it off so they can start chewing the other ones in. Because it's weird that they would take it off but not use it, right?
Yeah, it seems very generous.
That's the problem with the reductionist system of the gnotobiotic animal. Mm-hmm. You have to remember that we have lots of anaerobes in our gut.
Right.
And it's really a cooperative that's actually doing these things. It's, if you will, it's almost as if one microbe has one job and it does it, and then it gives the sugar to the other microbe, and we don't know what that other microbe is feeding B theta. And so it's a cooperation that's going on, and that may be my only criticism of a reductionist approach, is that you don't really see the, if you will, intact animal until they conventionalize the animal by giving it, complete stool, and then seeing the transcriptomics profile or the metabolomics profile that results.
Yeah, they do confirm.
That's a good point.
Yeah.
That's a very good point.
It's probably correct that they're probably liberating it, and then another member is catabolizing it, for example, and then BT is getting something in return, right?
And of course, the best example is when they began to work on methanogenesis. And of course, they had the pure culture methano something omlianskii, and they could never re-isolate it after they lost it. And they've been trying to hunt for that one methanogenic species for quite some time, and it's been lost to time.
All right, so now they start to use some bacterial genetics. They can make a strain of BT that lacks the sialidase, the enzyme that cleaves the sialic acid from the end. When you put that into your notobiotic mice, there is no increase in sialic acid in the gut, as you would expect. Then if you add S-typhimurium to those mice, the sialic acid catabolic operon is not induced, right? So there's no free sialic acid from the BT, so the sialic acid degrading genes are not turned on. Makes perfect sense. They also do some interesting experiments with another bacterium, Bacteroides fragilis, which has both sialidase and the sialic acid catabolic operon, so it can cleave off sialic acid and break it down. And when they put that in the gut, you don't get free sialic acid. So again, you colonize these notobiotic mice with Bacteroides fragilis. Now you don't get free sialic acid, so it makes good sense. So the idea is that Salmonella seems to be using the sialic acid that's liberated. They also show the same thing for fucose. They then make strains of Salmonella that are deficient in utilization of salic acid and fucose. They can't grow on these sugars in vitro, and they cannot compete with the wild-type Salmonella in the gut of mice, presumably because they can't utilize these sugars, which are being liberated by B-theta. Lovely set of experiments, really, really nice. Then they do a few experiments with C. diff. C. diff can catabolize sialic acid, but not fucose. And if they put C. diff into mice containing BT, the sialic acid catabolic operon is induced. Same experiment that we did before, looking at the transcriptome of C. diff. When BT is present, C. diff grows to higher levels, higher numbers, compared with germ-free mice. Again, possibly because the sugars are being provided to C. diff. The sialic acid catabolic operon, as I just said, in C. diff is induced in mice, but if you colonize the mice with a BT mutant lacking the sialidase, then the induction of the catabolic operon in C. diff is lower. If you feed these mice, all right, so you have a notobiotic mice colonized with a BT mutant that doesn't have sialidase, so the C. diff doesn't grow as well in those. But if you feed the mice free sialic acid, now the C. diff will grow better, even in the presence of this BT mutant, which can't release sialic acid, so that makes perfect sense.
And that was a great day in the lab.
Yeah.
When you got that result, right?
Yes, that's right. You can imagine, right?
Yeah.
So C. diff also depends on sialic acid provided by B-theta in this system. So far, the conclusion is sialic acid is an important substrate for these antibiotic-dependent pathogens, and then they go to conventional mice with a complex microbiota to see if this still holds. And it would be a bad day in the lab if it didn't, of course, because then you think, I'm looking at an artifact that only happens when you have one bacteria in the gut of these mice. So we have untreated mice. The sialic acid concentration is low, and they say this is probably because it's being consumed by the food web in the gut, the microbial food web. You treat the mice with antibiotic. These are the conventional mice. You see a peak in sialic acid in the gut, and that coincides with the susceptibility of these mice to infection with C. diff or Salmonella typhimurium. And then after a few days after you've withdrawn the antibiotic, the sialic levels start to decline as it's presumably consumed by the endogenous community as it's recovering. And in these mice, in these conventionalized mice, a Salmonella mutant that is unable to catabolize sialic acid or fucose cannot compete in growth with wild-type Salmonella. So showing that the use of these sugars is really important for growth of Salmonella. And finally, they looked at C. diff in these conventional mice. Again, they're antibiotic treated. The sialic acid catabolic operon of C. diff is induced. and a mutant of C. diff that cannot use sialic acid doesn't multiply to the same levels in the mouse gut because it needs the sialic acid. So the idea is that you get high levels of sugars after antibiotic treatment. This may allow a window for pathogens to expand. And then, of course, a number of other works have shown that this induces inflammation in the pathogens like that because it helps them to proliferate. And they end by saying, maybe this explains why one of the ways this explains why antibodies disrupt colonization. Can we use this now to make some therapies to somehow take care of post-antibiotic pathogen expansion?
So do they mean probiotics?
I don't know. So they don't tell us. So I'm wondering if you guys have any ideas.
So I had a great conversation with the two first authors yesterday.
Cool.
So Kat and Jessica are both senior graduate students in Justin Sonnenberg's lab. They're going to defend their thesis, each of them, this summer. I'll just as by way of background, Jessica, who hails from Baton Rouge, was able to start science as a high schooler. She actually did an honors thesis because each summer she was able to work on catfish pathogens at the vet school at LSU. And then Kat, she also was interested in science as a high schooler. She won a science competition, which got her an internship at an aerospace corporation. And then they each pursued science as undergrads and then graduate school at Stanford. But
nice story.
Yeah. So they were really excited by the attention that their paper got because it was a long slog. I'll just share with our listeners some of the backstory that happens with papers. We send it out for review. We get critiques back, and then we address them either with more experiments or with writing changes, and then you resubmit your revised paper. Well, they were involved
in this. You forgot the part about lighting candle at the cathedral.
Well, it was a year and a half between sending their paper out and then finally getting accepted because some reviewer thought that the beautiful experiment would be to make a mutation in C. diff that would block its ability to use sialic acid, and that would be a lovely proof of principle. Well, this is a lab that had never worked with C. diff before, and so Jess had to learn how to make mutants in C. diff, which is a lot harder than Salmonella or E. coli. So it took a lot of persistence. They both emphasize that Justin is just a terrific mentor. He's full of ideas. He's very bold, encouraged them to follow their own ideas, and is a great troubleshooter. And so I'm sure his enthusiasm really helped them through this long, technical, demanding period of trying to make a mutant in this new pathogenic strain. But in any case, it's a beautiful story, and they're interested now if they can apply this knowledge to not probiotics, but actually prebiotics. And by that I mean, if you anticipate that you're going to need a course of antibiotics, wouldn't it be great if we could give you a cocktail of bacteria that would make sure that there are no free sugars liberated that C. diff could then come in and grow off and replicate to high numbers and trigger diarrhea. Something both Salmonella and C. diff do.
Cool.
So that's
a good idea.
The kind of thinking that this work generates.
So they can test it in their mice.
Right.
Nice.
So what you would want to do then is rather than giving two drugs, is you would mix the probiotic as lyophilized event along with the antibiotic powder, and then you would just take one pill that would do everything.
That would be great.
Yeah. That's pretty neat.
Very good.
So this is very interesting, this idea. I mean, the C. diff sialidase, the sialic acid catabolic mutant, really is a nice addition, but it's not essential, I think. And this is what happens often with papers. They ask for experiments that take so long.
And that would be lovely, but you're right. They don't really
advance our knowledge.
It all that much.
Now, ASM has a journal called MBio where their policy is you don't ask for more experiments. If it's good the way it is, you can go. If not, you reject it and you try elsewhere. And I think that's good because this idea of making people do experiments, you can always think of another experiment, right?
Right.
So anyway.
Well, that's our job.
Yes, that's right. If we don't, we are out of jobs.
Yeah.
That's pretty neat. Nice story. Thank you, Michele, for talking with them.
Oh, it was great. And the other thing I learned is that we might have some competition from Stanford. They actually have a student and postdoc-driven podcast called Goggles Optional, where students and postdocs take turns discussing 2 or 3 papers that have just come out, and they also interview scientists. So Kat and Jessica were both interviewed when their paper came out, and then they also play a trivia game, so science trivia. And they too are pitching their science discussions for a broad audience, so trying to bring science to the public. So hats off to them.
That's cool. Goggles not needed?
Goggles optional.
Goggles, I have to check it out.
Yeah.
Cool. Maybe we can be on their show.
That would be cool.
Yeah. Why can't we be on your show?
We could at least take the trivia contest.
Nice. I like that. Yeah, I think it's cool that students and postdocs get involved in podcasting. I've had a number of them start up in response to our series and inspired by it and so forth. And I always tell them, let us know. We'll promote it for you. We'll help you.
Right.
So that's really cool. All right, let's do a few emails before we wrap up here. The first is from Jeffrey, who writes, Doctors, I just got around to listening episode 12, Photothermo Nanoblades and Genome Engineering. That's a long time ago. Your comment that it would need to be scaled up before it was practical for some of you to use intrigued me. I did a quick Google search, and I didn't see any recent articles addressing scaling up the process, though I'm sure that there are groups working on it. In my own amateur fashion, I'd like to suggest an approach to such a process. I believe that this is actually related to today's paper.
Today's talk.
Yes. I believe that technology applying microfluidic flow cells to single-cell streaming is reasonably well developed currently and improving all the time. If you installed the insertion capillary tube into a microfluidic flow cell and added a photosensitive trigger to the capillary tube, then it should be fairly practical to mass insert bubbles of materials into cells fed through the flow cell and collected on the other side. Although flow cells would most likely be tailored to particular cell sizes, it might be possible to make the insertion point slightly generic, i.e. slightly larger, by using laser forceps to automatically direct and immobilize the cells during insertion. The main restriction would likely be finding a cell culture that would support the microbe in question with sufficiently low viscosity so as to flow through the cell with ease while prevented undesirable cellular effects such as clumping or lysing while the cells are waiting to inject. High cell density at the input reservoir could probably be achieved by centrifuging the initial culture, then microdiluting up the resulting pellet. Although this may not prove to be the most efficient way to mass process cells, I do see another potential advantage in using a microflow cell. With additional solvent input channels before and after the insertion point, it might be possible to chemically manipulate the cell's immediate environment for periods of milliseconds to seconds in such a way as to make them momentarily more amenable to insertion without long-term consequences to cell survival. Such manipulations would be easy to study, cell species by species, in these flow cells until a manual of cells in optimum manipulation conditions could be generated. Anyway, thanks again for the fascinating look at research that I don't get a chance to look at very closely often or understand. I don't know, Jeff, sounds like you understand it.
It sure does.
Sounds like you have a future in bioengineering.
I check out San Diego.
Yeah, definitely.
I can't say that I digested everything. I'm going to have to sit down and read the letter again. But I think that some of what he says is certainly feasible and is being done.
Yeah.
But as I say, I have to really sit down.
Elio, you don't have a Salidase? Is that the problem? It's a joke. I guess it's not very good. Tarwin writes, hello, it's 25C in Palo Alto, California. I think humans, us, are so enthralled by the idea and stories of symbiosis because we see it as unusual. Even though our world is full of symbionts, life relies on symbiosis, we are blind to it most of the time until extraordinary examples are pointed out to us.
That's not true in the Midwest. We're very symbiotic out here. Friendly. Helping each other.
Okay.
You think you're different from the rest of the country?
Different from Jersey.
It sure is. People are very competitive here. Yeah. These make great examples of the precision, depth, and complexity of evolution and of life. It's easiest to make a headline in our mind when we see small examples of tightly woven systems than to keep the whole ecosystem of metabolism in our minds. Thanks, as always, for the amazing education and entertainment. Tarwin, who is a developer designer.
Well, I think there's a point to what he says. It's really quite possible that we're not trained to think about symbiosis being the default condition. We do see it as exceptional things, and we're amazed by stories we hear because they sound so unusual. But in fact, it's the opposite, just like Darwin says.
Yeah, it is.
Well, it's the paper today. The paper Vincent did is really a symbiosis where the good microbes are taking care of us, and when we kill them off or trim them down to below a critical threshold, invaders come in and disrupt the system.
And shit happens. Runny shit.
That's true.
It does. Wow. That's Midwest speak, huh?
I couldn't resist.
We don't do that here in New York.
I've never heard a New Yorker cuss.
No,
never.
No, we don't do that.
Right.
It's a Midwestern thing. We now, following in the theme of symbiosis, we have 2 emails about organelles and symbionts. So we had a little discussion about when an organelle is a symbiont or vice versa. So Jacob writes, just wanted to make a point about organelles versus symbionts. You suggested that the difference might be that if all the cells in the organism have it, then it's an organelle. However, not all plant cells have chloroplasts. Might the difference might the difference be just how high up the taxonomic tree the symbiosis goes?
Well, he's absolutely right about chloroplasts, and one should not bring that up as an example. However, the argument holds for mitochondria because although there are amitochondrial organisms like Giardia and a bunch of little things like that, they are thought to have had mitochondria in the past and have lost them. If that's true, then the statement holds that all eukaryotic cells have mitochondria. But except the ones that don't. So I think it's still a pretty good argument for mitochondria, but it certainly is not for chloroplasts. And if we said that, we were wrong.
Jacob is from Australia. And the next one about the same topic is Marion, who writes, TWiM 74, about 26 minutes in, the difference between endosymbionts and organelles is 1, when it happened, and 2, how much it increased fitness. The endosymbioses that resulted in mitochondria and chloroplasts happened many billions years ago and apparently conferred selective advantage to recipient cells, future eukaryotes, over non-recipients, future prokaryotes. We only have to wait a few billion years to see if the same is true for the endosymbionts discussed.
Sure. Yeah, I'm willing to wait.
Yeah? Okay. Let us know. And let's do one more here. This is from Bernadita, who writes, Dear TWiM team, thank you for all the great episodes. I'm loving every single one of them. I'm very pleased to hear more discussion on fungi-related subjects. Being from Lithuania myself, I wait every year for the mushroom season so that I could go and collect them. We even have national competitions for who can collect the largest number of mushrooms or find the biggest variety of them. Elio, maybe you should go there and collect mushrooms, huh?
I absolutely should. It's the many parts of Europe, especially Eastern Europe, where mushroom collecting is a rite. It's something that's done habitually and in great numbers, and so in some places they have special trains that take people to certain special forests. Absolutely.
On the mushroomy note, I would like to draw your attention to a recent paper describing the first fully sequenced genome of arbuscular mycorrhizal fungus. I think it's worth discussing. She sent a link to a PNAS paper. The genome of arbuscular mycorrhizal fungus provides insight into the oldest plant symbiosis. Oh, another symbiosis, huh?
Absolutely.
Am I pronouncing this correctly? Arbuscular?
Yes, you are. And that's an interesting point because the association of fungi with plants is said by some to have taken place at the very beginning when plants came out of the ocean and became terrestrial. So apparently you can find mycorrhizal associations in fossils that go way back. So studying the genome of one of these guys is certainly going to be useful and give some insights.
The second paper that caught my eye describes how Staph aureus intentionally induces pain through neuron stimulation in order to suppress the immune system's response to infection. I think it very much makes one think about systemic control of the immune system and, as ever, the unexpected ways that bacteria have evolved to evade it. And this is called bacteria activate sensory neurons that modulate pain and inflammation. And actually, I had seen this. It's on my list of papers I'd like to do here.
Oh, great.
On TWiM. So we'll do this in some future episode. It's a really interesting story. Thank you, Bernardita, for that. That's really neat. Enjoy I guess you're not in Lithuania anymore, but enjoy the mushrooms anyway. And that'll do it for TWiM number 76. You can find this episode at iTunes and also at microbeworld.org/TWiM. And if you like TWiM, help us by going over to iTunes. You can subscribe. It's free. And leave a rating for the show. It helps to keep us visible. And do send us your questions and comments to TWiM at twiv.tv. Michele Swanson is at the University of Michigan, which I understand is full of symbiotic relationships.
That's right. And
Some more symbiotic than others, huh?
Our basketball season ended, but golf season has just started.
Enjoy.
Thank you.
And thank you for joining us today.
My pleasure.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure also. Sure enjoyed it.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent. And Michele, the Masters is going on this weekend, so you're always welcome to come visit Aiken, South Carolina, where you can get a cheaper hotel room than Augusta.
And can you get me onto the course?
My old chairman religiously used to go, and so maybe. You never know.
You know how to reach me.
I know how to reach you.
Me too. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Chris Condian and Ray Ortega for their technical help. The music that you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.